Welcome to Microscopy 101, STEM program from Jump Simulation, University of Illinois College of Medicine, and OSF Healthcare. Really excited to bring this to you. I don't even know what I'm talking about. Welcome to Microscopy 101, Seeing is Believing. This is the first of 3 videos or recordings that are going to walk you through your brand new microscope and how to use every piece of it and get the most out of it. Microscopes are really kind of like a musical instrument. You know, they look kind of cool in the corner, but they're worthless if you don't play with them. So our big goal is to get you playing with the microscope, learning all the different parts, just like a musical instrument, learning how to make your own music with it, and really understanding and enjoying seeing things that you've probably never seen before, or going deeper into looking at things you've seen probably a million times before. We're really excited about that. We hope you enjoy all of the parts. My name is Shannon Egley. I'm a biomedical scientist. At Jump Simulation, I'm the anatomical coordinator, and at University of Illinois College of Medicine, I'm this clinical associate. I teach anatomy. Biomedical means that really my background is I love biology and I love medicine, and I really focus on that aspect of it. Here at Jump, I run the anatomical lab where we do a lot of surgical procedures, where we work with surgeons to give them practice so that when they do surgeries, it's not the first time they've done it, or if it's a really important surgery, which I think all of them are, they have had some practice, so they do it really well every single time. So they make all the mistakes in here where they can't hurt anybody, and then when they actually get to work with a patient like you, they do it perfectly. I'm also an anatomist over at the University of Illinois College of Medicine, so I get to work with those doctors before they're doctors, get to come over here. So it's kind of a cool background. The biology background, in college, I spent a lot of time taking biology, chemistry, physics classes because I love the way that the world works with us, the way that we work inside the world. And what's really cool is that a microscope is actually just like a screwdriver or a hammer. It's just a tool. And when you get really good at it, you can build a lot of things or create a lot of things. When I was in college, I had a chance to work with a lot of microscopes in a lot of classes. It didn't matter if we were looking at in chemistry, looking at something under a microscope, or in biology, looking at plants or animal tissues. It really didn't matter. Microscopes are a really important part of all areas of science, and hopefully through these courses, you'll get a chance to see all the possibilities of things you can do as a microscopist. So let's go ahead and get started. First thing you should have is a box that has things like your microscope and a packet of different experiments in it. But what you need to do is pull out your manual. So I actually have all of my pieces packed in this little toolbox. I found this toolbox at a hardware store, a real popular hardware store. It's all over the country, and I got it for $6. So, and it's perfect because it fits my microscope and all the tools. So the first thing you're going to need when we open this up, and I'll actually shift the camera here so that you can see everything. So there's the top of my toolbox. These are all the tools that you have, and we'll unpack those as we need them. This is a little extra space because I like to have lots of things. I'm really curious, and when I find something cool, like on my way to work today, I looked at this plant, and I really want to check that under the microscope, so I got that. And I also found a rock, so I'm going to put that in there. And I like hanging on to this for later. Like I said, some of those things you'll have. You see, my microscope fits right in here with the cover. And I put some balloon, packing balloons, whatever you want to call those, in here too. So I'm going to just pull out my slides and my microscope. The rest of this back in here later. Sit here on the edge of the desk. Okay, so the first thing you're going to want to do is pull out your manual. Oh, I'm losing focus. There we go. Pull out your manual. It's going to walk you through everything. Yours like this. You can 3-hole punch it and put it into a binder or do whatever you like. But this is the manual. So let's go ahead and go right into it. I want to make sure we're playing as much as possible, not talking all that much. So first page, think like a scientist. I really think that this is an important thing about everything in life. It doesn't matter if you're working at McDonald's or if you're an actual scientist. Thinking like a scientist is telling you to keep questioning things, keep asking questions to understand the things around you, to do whatever you're doing better. And it's really important. When I think of fast food restaurants, I don't want to say any names, but I've worked in them. And what's interesting is that a lot of times when we go into the job, we just do what we're told because we want to get a paycheck, but we don't really ask why we are doing what we're doing or get curious about why we're doing what we're doing. So what's crazy is that you're born to ask questions. You're born to wonder why you're doing what you're doing. You are born to get better and better because you are curious. So the best scientists on earth are probably babies. And if you get a chance later, there's a QR code down here on the corner that has a little short study on different things that babies do that are like a scientist. But when you're first learning about the world, you don't just come into the world and know everything. In fact, you can barely use the tools that you came into the world with, like your eyes and your ears. Those all have to be trained on what to do out in this environment. So what you want to do as a baby is you grab things, you put them in your mouth, and you taste them, and you smell them, and you listen to things. You listen to the noises around you. You hear everything, and you're just taking in the world and understanding everything. But a lot of times as we get older, we stop asking why or exploring or wondering about things because we get to this point where we've asked so many questions. People around us say, just do it because you're told to do it. Well, if you're at a burger joint, you're getting a paycheck doing what you're supposed to be doing because they told you to do that. But what if you ask some questions and you're doing something one way and it could be done better a different way, but nobody stops to ask the questions. Never just assume that everything's the way it's supposed to be and it's never going to change because things do change. It reminds me of the story about a guy that his family was at Easter time, which is right about now, and grandma and mom and great grandma are all in the kitchen in different areas and working on different things. And he said, saw his mom slice the end off the ham and throw it in the trash. And he was, why'd you slice the end off the ham and throw it in the trash? And she said, well, because grandma did it that way. And so he went over to the other side of the room and said, hey, grandma, why do you slice the end off the ham and throw it in the trash? And she says, well, that's what my mom did. And so he said, well, I'll go ask great grandma. She's in the other room. So when I asked great grandma, who was relaxing, and he said, why do you slice the end off the ham and throw it away? And she said, oh, because my cooker's too small. So probably 40 or 50 years ago, when grandma was making Easter dinners, she threw away the end of the ham because she couldn't fit the whole thing in the cooker. And that was the only way to do it. But of course, now we have bigger cookers. And what's bad is that everybody had been throwing away that piece of ham, that big end section of the ham, because of something that happened a long time ago. Nobody questioned why. They just did what they did because that's the way it's always been done. Don't do that. Question things, explore, wonder about things, be inquisitive. Look at things like the QR codes. You also want to have somewhere that you can write things. So we've left some blank pages here if you want. The scientific method is actually something that over 1,000 years, scientists or anybody that's observing, doesn't matter if it was back at Socrates or Galileo or whoever, ask questions. And we found that there's a certain method or a way that you can write questions to make it most efficient. And the first step is just observing. People notice things and how you behave, how the world around you behaves. Like, for instance, people keep going in the doors behind me. Dang it, let's stop doing that. Poop. Oh well, practice run. Right. Scientific method. And sometimes when we talk about research, ask yourself, why is it like that? You go through these steps. What can I do to make things different? Let's say you're making Jell-O or whatever. You notice that the Jell-O is not jiggly enough. So you observe it's not jiggly enough. Well, ask a question. What can I do that's different? Can I put less water in it? Can I not use fruit? Can I use fruit? Question it. Form a hypothesis. Well, I noticed that when I put fruit in my Jell-O, certain fruits, it stays watery and other times it doesn't. So pick a couple fruit, design an experiment. Let's make a bowl of Jell-O, 2 bowls of Jell-O, 1 bowl of Jell-O, 3 bowls of Jell-O actually. 1 bowl of Jell-O is just Jell-O. We call that the control. Another bowl of Jell-O has pineapple in it. And a third bowl of Jell-O has grapes in it. And then you run the experiment. You make it, you notice that the controlled Jell-O is the way Jell-O is meant to be. Look at the grapes. How are they compared to the original one? Were they more watery? Were they more stiff? What happened? Look at the pineapple. Was it more watery, more stiff? What happened? Collect your data. What happened in these 3 experiments, and then change it, modify it. So maybe you notice that the pineapple Jell-O is more watery. So what do? See what you can, how you can change it. Can you add more water or less water the next time? Do something different. Experiment, repeat, modify, and do it again. By the way, it's actually a fun experiment. If you have a couple boxes of Jell-O, try that. Pineapple in one Jell-O, grapes in another Jell-O, and just plain one. Next thing you want to do is you want to have the right tools. And the first tool is actually with you all the time, and it's right between your ears. It's your brain. Be inquisitive, ask questions, wonder about the world. What's interesting about wondering and asking questions is that the more you wonder and ask questions, the more you wonder and ask questions. So if you come up with a couple questions every day, or you do something a little bit different every day, and you do it every single day, you'll notice that you do it more and more and more. Your brain is trained to do things that you do a lot of and do it well. So if you ask questions now and you're not maybe so good at it, do it again and again and again. The more times you ask questions, the more questions you come up with, the better your questions are going to be. So every single day, I would just mix it up a little bit. Albert Edison, Thomas Edison, it holds most patents for creations or inventions. There's a long story behind that and a lot of debate, but he did have one thing that he did right, and he had what he called the Edison quota, is that in a week he had to come up with a certain number of questions or a certain number of ideas. It didn't matter if they were great ideas or they were flop ideas or dumb. So it didn't matter if he was sitting and staring at the light bulb that he invented and went, hey, what if? Or if he was brushing his teeth and said, hey, what if? He would come up with a question or an idea and then go with it. If one of them panned out and seemed like it was something realistic, he kept it. But he also did the same thing for his employees. And well, that's a lot of debate, but have the right tools. You have the right tool, but your tool in between your ears is just like a muscle. You have to flex it, you have to use it. The more you flex and use it, the bigger and more powerful it gets, just like a muscle. Right? Next thing we're gonna do is we're gonna look at microscope safety. All right? I'm gonna walk through these and we're gonna pull the microscope out because it's really important to understand the safety procedures for the microscope before you even start using it. So the first one, always work within shouting distance of an adult. Always make sure that there's something around that you can yell for because you're working with glass and very thin glass. If it breaks and you cut yourself, you want to make sure you have help right away. It's always good to have Band-Aids. So you might want to write that down in your notes over here somewhere. Get my pencil and do it along with you. There's my pencil. Okay. So some of the extra things you might want to have are Band-Aids, just in case. You want to keep trash or Sharps cup or paper towels nearby in case you spill or drop something. So another thing you want to do is have a Sharps cup. And it can be something simple as a disposable plastic cup. Because if something breaks, you want to be able to put it in there. If you use glass slides and you're done with them, you can put those in there too and keep them safe so they're out of the way. One of my biggest peeves is actually using something sharp and not knowing where it's at because I've cut myself on accident. Just by sitting something down in the past and forgetting where I put it. All right, never eat or drink near the microscope because things that are microscopic that you can't see can actually get on the food or the drink and then you put it in your mouth. Even if it's a little chip of broken glass or an edge that breaks off and gets on your finger. If you're eating a sandwich, you can easily put that glass into your GI tract and that would be really bad. All right, handle chemicals and specimens with care. If using potentially dangerous or infective agents, wear gloves and glasses. So if you're using something that's infective, make sure you have gloves. If you're using something wet that can splatter, other than just plain water typically, you want to have glasses on. Okay. If you're wearing something that spill or you're working with stains, maybe you want to wear an apron, something that can cover you up. So make sure you have protective equipment when you need to. Everything that you have is safe to get on you. Iodine we actually put on our skin to kill bacteria. Methylene blue, you don't want to taste it, but it's actually safe for some reason. It got in your mouth. But you want to make sure that you're always thinking safety all the time. Next, keep 2 hands on the microscope all the time, one on the arm and one on the base. And I'll show you in just a minute what those 2 things are. So the arm and the base, always 2 parts in case there's something loose and you don't drop your whole microscope. Okay? Keep the scope covered when it's not in use. You can see even when mine was in the toolbox, it was covered. Okay. Always start with the stage furthest from the lens, and I'll show you what that means as we get to it. But basically, there's a way that as soon as you uncover this and before you put anything into it, you want to set the knobs appropriately. And I'll show you that walkthrough. Never take your microscope apart. There are a couple times that I'll show you how to take things apart, but in general, don't take it apart and try to put it back together. I know I was telling you be inquisitive, but in this situation, this thing is a precision machine. It's like your car at home. If you don't know what's in the engine, you don't just rip your engine apart. So the pieces that I'll have you take apart, I'll walk through one step at a time with you to make sure we get it, right? And then the last one is really important too. Never use anything except lens cleaning tissue to clean your microscope. You have a pack of lens cleaning tissue, and it's really important that you use this and not your shirt or a paper towel or a Q-tip or anything else, because lens cleaning tissues are 100% grit free, which means there's nothing on them. They're like perfectly clean. Right? Even something called a Kim wipe that I have in the lab that I use a lot of, like this, even these tissues are only 80% grit free. I use these on my glasses, but my glasses aren't a powerful magnifier. They're a mild magnifier, and we'll talk about magnification later. So when you're cleaning your lenses, any lenses on the microscope, use this, just this. If you run out of this, you can go to a camera store and get more. You can order it online from any science place, you know, Amazon, Google it, whatever. But you want to make sure you use these. They're not very expensive, but these are dedicated for microscope lenses or special magnifying lenses. So you want to make sure you use those. If you use any cleaning solution, you can use 50% Windex. So I bought lens cleaning solution that's made especially for it by actually one of the original microscope makers, Zeiss. And so this goes back a long way. And I think I got this at Walmart. So it was really cheap, a couple bucks for the whole pack. Remember, don't use anything else in the pack except this. When this goes empty, you can use 50% Windex, 50% distilled water or clean water in it too. Right. So let's move on. Let's unpack this kit. The best part. This is your lab. Okay. Again, you have some notes over here, space over here, so you can take and jot things down. You are going to pull your kit apart now. So you got a box in the mail, and I'm sure you've already done this, but you want to pull that box out. Microscope in it. Here's your box of goodies. And then carefully sliding it down, pulling it out. It's all taped up and safe in here for now. But you'll slowly cut around the edges, or you can grab the edge of the tape and peel it all the way off. And then when you open it, put it down flat, peel it open like a book, and your microscope's inside. All ready for you to pull it out and start working with it. You also have a box. Everything's in this box, plus everything that comes in this envelope is going to be in this toolbox, so I'll be unloading it from there. So here are some of the basic tools you're going to get. In this envelope, it has things like extra tools that don't normally come with a microscope. It'll have a bunch of envelopes like this that will say Microscopy 101 on the top, or 102, or 103. And this has all the experimental materials that you'll need for that experiment. Okay. And walk through the pieces in your microscope. Right. First, your microscope cover is folded up into a little tiny square in that white box, but this is your microscope cover. It's important to use it no matter where you put the microscope, even if you put it inside the toolbox, tackle box, whatever thing, or you put it in the cabinet. Always make sure that your microscope's covered all the time. Dust is really bad for microscopes because dust is dirt. It's microscopic particles that can actually scratch a polished lens. So you're looking at something that's a very delicate machine. When I said earlier, hold it with two hands, one on the arm and the base. Your first anatomy of the microscope is this is the arm. The arm has a handle on it. It's waiting for you. And here's the base down here at the bottom. The base is what sits on the table. Okay? So when you carry it, always hold it by the arm and the base, just like this. Always. Okay, so if something slips or something falls off, you've got a good grip on the majority of the microscope. Put that down. I'll actually slide these things out of your way. Right. So microscope, you have a lens set. This is called a Barlow lens set. And normally they're in the package, but I was playing with mine earlier and I just sat them in here really quick. Right, so you open these up. You have one that says 20x, 20 times magnification. You have the actual tunnel, Barlow lens, and we'll talk about this later. And then you have, whoops, I have two 20x's. You have a 10x also. I'll just put this down for now. My 10x is actually in my microscope over here. Okay. You have a phone mount. And it was just like this. You don't have to use the phone mount. The phone mount is just handy if you want to, if you don't want to put your eye straight up to the eyepiece and look all the time, or if you want to take pictures, you can use this to mount. I'm having a problem unwrapping it, so I'll just stick it right here for now, because I already have the one I'm going to use over to the side. This back. Down here, so we have a little more room. It came with a prepared slide set. When you open it, one good thing to do is somewhere on it, maybe even on the front if you want to keep it clean and tidy, just right top. I hope it never happens to you, and by do it right on top here, hopefully it never will. When you're going to open this, make sure that you know which side's up, which side's the top. So I'm going to open this up. You can see, keep that foam padding in. It helps hold the slides in place. Here's a list. What's on all the slides. We'll talk about how that list works and what the initials all stand for. Okay, there's a slide box. Take care of this because the slides are thin and very, very breakable. So I'm going to put mine over here. Microscope cover. My actual microscope. Here. Blank slides. You have another little box. It's like this, and you see I already wrote top on the top. This out of the way, see if that helps with focus. Yeah, it says top, same idea. If you open this upside down, oh, bad news. All these slides will go all over the place. So in here you have another piece of foam. You have 5 prepared slides in front, and then blank slides in the back. You'll also have a little box of slide covers. Okay. And we'll pull these apart when we need it. It's a little tiny box. It won't fit in here. Please don't try to fit in here, because if you try and cram anything in here that doesn't need to, you could break one of those slides. Right. So we have that. Oops, right here. Slide covers. Okay. Next, transparency paper. And it's exactly that. It's just a piece of transparency paper. Back when I was in school, our teachers would write on these and it would project wherever they wrote. They would have a projector that projected wherever they wrote up on the wall. You're going to use this because you can actually cut little slide shaped, pieces of plastic off of this, so that you can use a cheap or inexpensive way to have slides, because glass slides can get a little bit pricey, but this is really cheap. You'll also notice down in the bottom corner, there's a printed area. I'll show you how that works. It looks like it has a little measuring device. We'll do that. Just gonna slide this to the side for now. Next, you have ring stickers like this, or maybe just a little bit different, and that's fine. Another one over here somewhere. Because I actually changed where it has the insert in the center still, so you can use those. I'll show you how to use the inserts even later. These are cool little donuts. We're going to prepare wells on our slides with that. You have 6 vials. 1, 2, 3, 4, 5, 6. And we'll, as we need them, we'll explain what each of these are, like gum media. Specimen vial over here. You should have a little vial with shrimp eggs, sea salt for their water, so when you grow them. A little packet of yeast, and then methylene blue and iodine. You have a small hatchery. Looks like a teeny tiny petri dish. So when you think of scientists having that glass dish they grow bacteria in, that's what it looks like. Right? It's actually a hatchery, so you'll put water in this and then grow your shrimp in it. Lens tissues, I already showed you those. Tweezers. Forceps, pickups, tweezers, whatever you want to call them. There they are. A pipette. Also known as an eyedropper. We'll use that. We'll use all of these and show you how everything works one at a time with each experiment. You'll have a paintbrush right here. A spatula there. Probe or a pick looks like this. Pencil. Seems like a simple tool, but it's still a tool. A marker. You'll actually have a marker pack just like this. It has your marker in it. Also has a ruler. Wondering what are we going to do with that under a microscope? I'll show you. And it has some papers that you can use for marking your specimens. Toothpicks. And these are just run-of-the-mill toothpicks, so if you use them up and you need more, you can get it anywhere. Dollar store, you can get 500 for $1, I think. Pencil sharpener, which we're going to use as a microtome, and I'll show you how to use that. Be careful, it does have a sharp edge. And we're going to use it for more than just a pencil sharpener, too. So that's my favorite thing to do, is find multiple uses for different items. Clear tape and nail polish. So clear tape, we can use this juice to make slides. Nail polish, preservative, something to put specimens in that can keep them in the same condition for months, potentially at a time. Microscope cover, we already did that. 3 AA batteries, or sorry, AA batteries, which I've already put into the microscope. You can see them there. Shifting a little bit. First one. Second. Three batteries. And your lab notebook, guides. So your handouts look like these. So you can 3-hole punch them. I suggest you get a notepad, kind of like a journal. So your notepad or your journal, you can keep all the notes you possibly can think of in there. And we'll actually talk a little bit about that in a while. And then the microscope manual, which is the last item. And it's the one that came with microscope. Let's place it already. That didn't take long. Well, it's a manual that on the front of it has a picture of your microscope, and inside of it has kind of another section of how to take care of your microscope, what you can do with these specimens. I'll show you how to use all those too, and kind of gives you an overview of some fun things you can do with your microscope and some cleaning tips and caution tips. So that's your set. Everything you need, all handy dandy wrapped up. And also, as we go through this, we're going to walk through everything. Don't jump ahead. Don't lose focus on what we're doing. No pun intended with the focus bit. But as we go through it, I will show you how to use everything. So don't jump ahead. Make sure you follow. If you need a break, you need to think about something, or just use the bathroom or breathe or whatever you need to do, go ahead and just hit pause and and come back to it. Okay. So the next thing is really important, and this is collecting and culturing. This seems like it's kind of really quick, but we're going to be doing some work with some live specimens, so we really need to let them rehydrate. When you're working with live specimens, there are a couple of things to think about. Number one is if it's actually living and you can set it free, please do. One of the bad things about microscopes is that most things that you look at actually have to be dead. It can be something that's already dead and you pick up on the ground. One of my favorite things to look at actually is if you look in your windowsill at home, there's usually a dead ladybug or a dead fly or a dead mosquito or a dead something, a dead spider in there. Just take your tweezers, pick it up, put it on a slide and check it out. If you see an ant crawling around, I actually saw this yesterday and thought, oh, I need to find some time to do this, but I'll show you how to make a small slide that you can corral the ant into, and you can look at the ant while it's alive. We're also going to have some ants that are, or bugs that are in your prepared kit too. But when you're doing this, you can find specimens everywhere. They can be living, they can be from your refrigerator. There's a lot of living things in there you don't realize, like in yogurt. Just wait until you get a chance to take a peek at that. But specimens sometimes take time to prepare. So what we're going to do right now is actually prepare some specimen. First thing you'll want is your little specimen tray, your hatchery, petri dish. Okay. And this is nice because it's 2 containers right now. Right. When we set up an experiment, we always want to remember what we're working with. So you can either take a page out of your manual or a page out of your notepad or a Post-it note, and on it, we're going to rehydrate some things. So I'm going to put these 2 items down, and on one side I'm going to write lichen, L-I-C-H-E-N, which is the fungus we're going to talk about that grows on trees. And then the other thing we'll look at is moss. Right. So I have my specimens marked, labeled. I know what's in them. Okay. Just in case something gets mixed up, I might write big for the container and small over here, just in case I'm not paying attention and someone comes along and moves my things around. I know which is which. I like that. Another good idea is actually taking your marker and writing on the bottom. The problem with the marker is that it's not going to come off very easily. So if you can put a piece of tape on the bottom and mark it, that's great. Take a piece of your Post note, write on it, flip it over and mark it. That's great too. Where you can get specimens, you can get specimens everywhere. Your back porch step is actually a universe for microorganisms. So you can take a scraping off the porch step. You can go and take a little sample of water from a pond or a river. I actually did that. So I have a little water bottle with some pond water in it. Rain puddles that have been sitting there, you never know. You know, even dirt that's in the rainwater has microorganisms in it, and they get stirred up when the water comes around. And we'll teach you later how to agitate soil and introduce some water and see what's in there. Tips: make sure that whatever you're using is clean of any cleaning products, pure of any cleaning products. So make sure you rinse these off before you use them. When you're testing different things, even if it's your backyard, test different areas. If it's a tree and you take a scraping off one side, take a scraping off the north, take another scraping off the south. Look at different areas because you'd be surprised. Look at different depths. If you're going to the pond, skim some water off the top and look at it. Skim some water from way down in the bottom and look at it. Right? So I've actually put a QR link here on some different examples of how to grow specimens. We did a STEM activity where we grew specimens on cheese. So it's kind of cool. If you have any cheese in your refrigerator and your parents are okay with you using a couple slices for experiments, go for it. My little tip is that if you want to experiment with things that are on your hands, experiment. That shows you how to put your fingers on, how to clean your fingers and all that. You can put it on any part of you. If you want to put a piece of cheese on your face, leave it there for a minute and then pull it off, see what's growing on your face. A lot of fun stuff you can do. So we're going to get our live specimens going right now. So the first specimen will be a little piece of moss. And that's in your envelope. So if you want to pull out envelope number 1, in the corner. My Crossbee 101. Inside of it, you'll have a little piece of moss. Just like this. And you don't have to use the whole sample. You can use part of it. You can see it's kind of falling apart. We're going to put the moss down here on the bottom. And I should probably be using my tweezers. A much better idea. In fact, I'm going to take a couple of these loose pieces, tuck them down. The other sample I'm going to do is the lichen. So you can see the lichen. This is a piece of bark off a tree. It was dangling there, almost about to fall off, and I popped it off. I typically prefer not to do that, but if it's laying on the ground, like for instance, I have a piece of the tree here somewhere. When I re-record these, I need to make sure my cable is free and clear. But I found a branch actually in my yard that was covered with lichen. So what I'm going to do, I'm going to take this, I'm going to scrape a little sample off. Yeah, this looks really good. Wow, that had a hole bored right in it. Uh-oh. See a little hole here? So I'm going to scrape some of this. And I'm actually going to break a small piece off. There we go. All right, so I scraped some of the lichen. I actually have some of the tree bark itself. I'll put that right back over here. This is why you wouldn't have a garbage can close by, because you make a lot of mess. So when you're rehydrating things, there's a lot of microorganisms that are growing on this. When they get really dry, it's called desiccated, they get dried up. A lot of microorganisms protect themselves by wrapping themselves in a little protective covering, and it's called a cyst. So it's like they go into hibernation mode, so that when it's really dry and there's no food or there's no moisture, they can actually not use a lot of resources. But when they get wet again, they'll start cracking back out of the cyst and they'll reanimate, they'll come back to life. They weren't actually dead, but they'll be more active again. So keep in mind, these are dry environments. If you find some pond plants, those are great to use too. But I'm just going to use, this has been sitting at room temperature, some water. I'm just going to put enough in here. Oops. So my sample is wet. The bark's nice because it's floating. And the same thing here with the moss. Okay, I don't want to drown the plants, because the organisms are used to having a dry environment, breathing air. Remember, just like you, if I hold you underwater for a long time, it's going to be hard to breathe. Okay, so we're going to rehydrate this. I want to make sure that whole moss is wet, so I'm going to flip it over and kind of drench it. Oops. Add a little bit. I feel like I'm doing a cooking show. I'm just going to pat the moss, pat it down here. Moisten. So I've touched with the tweezers the moss. What I don't want to do is go over and touch the lichen. So I'm going to put my tweezers down right here, and I'm going to use my pick for the lichen. I'm just going to push it down. I'm going to make sure that's good and wet too. Can you think of why I didn't want to take my tweezers and use it in both specimens? This is called contamination. I don't want to contaminate my bark specimen with my moss and vice versa, because if I'm looking for specific things in this, I don't want to introduce them over here. These are microscopic. They could be already clinging on the tip of the tweezers or on the tip of the probe. So I want to keep my tools separate. Another good thing that you might want to have handy, you can go back here to the handy dandy things you might want to add to your lab, rubbing alcohol. It's great for cleanup, even those alcohol wipes. But once you clean with them, it kills off most of the microorganisms. You know, like the Lysol wipes say 99.9% of the organisms that are living on there are dead. You know, germs, I think they say. Okay, so we've rehydrated. We're gonna let those sit there, and in about an hour or so when we're in our next segment, we'll come back to these and we'll actually look and see if we can find any microorganisms that are reanimated in here. Soil ecosystems, doesn't matter. One teaspoon of soil or sample like this, you'll have hundreds or thousands of organisms growing in there, living in there. So it's their own little universe right there that they'll never go beyond. It's like you in your hometown. They're stuck in this little area. They're not gonna go anywhere else. They stay there unless they hitch a ride from somewhere else. So later we'll actually talk about how to rehydrate soil, which is pretty easy. We have a whole module actually on soil and getting it damp and filtering it and looking at it and teaching how to do that. So this is just an introduction, but you can look at soil through the QR codes. And we'll do the same thing with activating yeast. We'll do that later when we need it, because we can get something good from yeast with only like 10 minutes worth of time. But with this, we want to make sure this may actually take over a day to get all the microorganisms active again. So it's something we'll look at in a little bit. You can revisit it later today or tomorrow or the day after. Usually within 2 days, then you want to get rid of your specimen, right? So here's kind of an overview of the different lessons we're going to be working on. Always keep your mind open and think about, the possibilities. Like I said, be a scientist, be curious. So think of things like weird things, like why do you stink? Why do you get armpit odor? Why do you get foot odor? Why do you get bad breath in the morning? What's interesting about bad breath is that the organisms that are living in your mouth during the night, normally during the day, you keep your mouth closed, right? But at night, when you snore or lean back, the air is really going through your mouth. And so those microorganisms, the bacteria that love air, are getting the feeding, they're enjoying it, and they make bad, stinky smells. So maybe you can look at different bacteria that's in your mouth sometime. Or look up what kind of bacteria in your mouth. What can you do to try and get rid of that stinky morning breath? Is there anything you can do? You ever like drank milk before bed and now you've given that bacteria a lot of sugar and fats and now that bad stinkiness. What's interesting is that you never know when you're going to find something really interesting just by being curious. Like a doctor that actually found out that bacteria growing in your stomach can cause ulcers and give you halitosis, which is really bad breath all the time, not just in the morning. And nobody believed him. People were, even just a couple of decades ago, people were thinking, oh, bacteria, schmacteria, it doesn't do as much. It's not a big deal. There's no way that bacteria can live in your stomach because it's so acidic, and we'll talk about acids later, but it's really hazardous. Most bacteria that goes in your stomach are killed. This guy was very firm. He's like, I know this is it. So he took samples from people that had ulcers, constantly bad breath, had ulcers, which are holes eating at the stomach lining that make a lot of pain. And he found that there were some things that were living in there, bacteria. And he found that these people that had these problems had similar bacteria. Nobody would believe him. So you know what he did? This is gross. He ate the bacteria. Yeah, so he ate some of the bacteria on his own. He waited, and sure enough, he started developing really bad breath. Then he started getting really heartburn. He developed ulcers, and then that was his way of showing this bacteria causes these problems. And about 9 out of 10 people that have stomach ulcers or halitosis, they find have this bacteria. So that was his way of going through the scientific method. Right? Another thing you want to think about is don't experiment on yourself. Don't experiment on other people. He took a big risk. In fact, he caused pain and suffering to himself, and that's a really bad idea to do. But he discovered something. And how do you get rid of bacteria? If you get sick and it's because of a bacterial infection, what do? You take an antibiotic. And so what's interesting is that these people that were on pain medicines and other things for the pain, now they take an antibiotic just like if they have a really like pneumonia or bad cold or other kind of infection. They ate these antibiotics that went in and killed the bacteria in their stomach, and poof, miracle cure. So you never know when your curiosity is actually going to make a difference. That QR code, I've tried to put a lot of QR codes in here so that you can, take breaks and go off and do other things. So the first tool that was important was your brain. The second tools for being a microscopist are the microscope tools. Your lab, this has everything you really need to get going and be able to explore a lot of things. If you get really good at this, there are a lot more stains you can buy or a lot cooler microscopes you can get, but your microscope is actually a really good one. We'll walk through it in just a little bit. But the third tool is a notebook. And, you know, as far as priorities, I would say the notebook comes really close to the number one with your brain, because your ideas, your thoughts, you want to keep them in a notebook all the time. I learned a long time ago about journaling, and I got my daughters into journaling, and my daughter gave me a really cool journal that I actually have over here to the side. But it's a good place to store your ideas. You know, it doesn't have to be like your diary secrets or whatever if you're afraid of people learning what's in your head, but just the cool things you think of, or the things you've experimented with and you've discovered. Your notebook is really, as a scientist, like a recipe book. So it's a cookbook. When you do something, you write down all the steps that you did. The first thing I did is I pulled some bark off a tree, right? Second thing I did is I hydrated it. A little more detailed probably, but it's all of your ingredients, all of your methods that you use to do anything. Through this, I put a lot of recipes in here, but I don't want you to just look at my recipes. It'd be great if you had your own notebook that you put in your own writing. So a special way that you do those recipes, so that anybody can take your recipe book and repeat it. That's science. Science isn't just doing things for funsies. Science is doing things to learn about it and to share with others. So someone else can take your notebook and they can follow your directions and actually do the same thing and get the same results as you. That's what makes a good quality experiment, is repeatability. Your recipes have to be repeatable. You know, maybe you know somebody else that's in the STEM class, you know, somebody else that has a microscope at home, swap some ideas, you know, find some plants in your neighborhood, write them down, put a little sample in here and rip it out and hand to them, or make a copy and do something fun. Get creative with it. Make a social network of microscopists of your age and your friends. But the number one thing is document everything. It doesn't matter what it is. If it's a fleeting thought and you're thinking squirrels, oh man, you know, a gray squirrel, I wonder if gray squirrels are like rats. You know, whatever it is, write down in the corner, write it down. Thoughts are like dreams, right? You ever had a really cool dream? You wake up in the morning and go, I got to tell people about this. You eat breakfast and you're like, what was that dream again? I don't remember. So I used to be a musician. I loved, you know, writing and playing and creating things with my guitar. And sometimes I'd wake up in the middle of the night with this cool dream, this melody in my head, and I had to sit down and write it down. So I would actually walk to the next room, grab my notepad and write it down. But you ever had that cool idea and the next day you're like, everybody's gonna love this, and then within 15 minutes it's gone? Okay, that's what notebooks are for. Write down all of your thoughts, all of your ideas that you think mean anything. It doesn't have to be about this subject, it can be about any subjects, right? Right. Make sure you're writing note pages so that you can reference them and make a table of contents. Your notepad, and I should actually probably put something in about notepads here, but is important for your thoughts. Your brain can only hold so much in your active memory at a time. So it's good for recording things that you don't have time to explore right now. Keep a broad mind. Think outside the box. Think creative, right? And what else can I do with that moss or that lichen? Da Vinci had 7,000 pages of notes and over 7,000 pictures in his notes. And they weren't just of like the Mona Lisa or his flying machines. They were everything you could think of, anatomy, how to make a better paint when he painted The Last Supper. If you've seen that, I'm sure everybody's seen that picture. But The Last Supper, he was creative and he tried new paint formulas that he thought were going to last forever. He was wrong. That painting, that mural actually, is fading and falling apart, and it's hard to keep that. But he was curious and explored, and at the time it looked like it was going to hold up using like egg whites and stuff in his paint. It didn't with time, right? But that's the thing. He experimented. He wrote it down. We know what he did, and we've seen the results decades, you know, thousands of years later. Right? The invisible world. Here's what you're here for: to see things you can't normally see. Keep an open mind. Be curious. Don't forget to ask why constantly. The other way I want to think about this is think critical. So critical thinking. Okay. The thing about being wise or understanding a lot is knowing there's a lot you don't understand and a lot of things you don't know. But exploring and asking questions is a big part of that. Okay. It doesn't matter if you're going to be a scientist or a doctor or a nurse or a geologist or an archaeologist. It doesn't matter. You have to think critically, think and evaluate the things that are around you. Right? At one time, you know, we thought that people got sick because of the air, not things in the air, but the air. Stinky air can make you sick. It was called miasma. So you breathe it in, and oh my gosh, now you're sick because of it. Now we know it's germs. There's a really famous study that I actually put a QR code to. Is John Snow, probably one of the first epidemiologists. We're in a pandemic, which means this health hazard is going around the world. An epidemic is across the nation, our continent. But John Snow in London was finding that cholera was all around him, all over the place, and people thought it was because of bad smell. So you're around dirty people, smelling them, and that's how you got sick. So people avoided different classes, and it was very bad. But what John Snow found is he took a map, and he looked at the map, and he said there's got to be some central cause. And he looked at where people went shopping for their groceries, and where people went and socialized, and where people went and got their water and things like that, because there was no running water back in the day. It was a hand pump. And what he found is that most of the cases, and he would put little marks on a map of the cases of people that died in different severities, and he found there were some central locations to these deaths. And he thought, what is the one thing in common in this area? And he found a well. There was a well where people went and got their water to get back to their families, and a lot of those families got sick. He talked to the church, and church and science didn't always get along back then. And so it was really nice because the church said, this is what we're going to do. We're going to work with you, and we're going to do what you think will stop this. And he said, we need to take the handle off the pump, because they'd have to crank it, no electricity, right? So they would have to crank the pump and get the water out. So if you take the well handle off, no water, they'd have to go to other wells. And that well handle was kept in the church, and it's actually framed now because it's so famous. But people stopped going to that well, and they started getting better. And that told them there's something in the water. They didn't know what it was yet, but it was something in the water that was causing this. And nowadays we actually know cholera is formed by bacteria called Vibrio cholera. So we know what it looks like. Microscopes are what let us know. So you'd take water from that sample, you'd look at it in a microscope, you'd stain it, you do whatever you have to, look for the unique features to that one sample of water, and you could find your killer, right? So think creatively, think out of the box, think of reverse engineering, taking the big problem and breaking it down into little pieces. How could it, here's what our problem is, go backwards. Where could it have come from before that, and just keep breaking it down, right? So when you think about it, think about size. This is a really, my QR code is missing, but there's actually a link to different sizes. Maybe it's on the other page. Nope, I have to add this link. QR for size exploration. But you can actually go to this website, and it'll let you look at a human, and then zoom in and zoom in and zoom in, and you can get an idea of how small things are, all the way down to a water molecule, all the way up to a planet, actually all the way up to the universe. But think in perspective, how small can you see? Make a note. What's the smallest thing you can see with your naked eye? Can you see a pencil tip? Can you see something smaller? Is there something there? How about cancer cells or bacteria or viruses or individual pieces of sugar or water? Get curious. Complete history of microscopes. Hold on to your bootstraps. We're on for a good trip here. So the complete history of microscopes is not going to be that detailed. It's really, how did it start? And it was originally started, magnifying glasses. People taking a piece of glass and just looking into it and seeing magnification. You can do this if you take, any of these, right? Here are your lenses. You can see there's actually a glass lens right there. Looking at it, that easy. Let's flip around. Oh yeah, very nice. So looking at paper, magnifying, we've been doing that for thousands and thousands of years. I think cavemen probably did it with little pieces of crystal, right? What we can look at, that's not true, I don't know that. We can look at that and magnify it. It's really cool. But some people wanted to say, how close can we get? So what's really interesting is that a fabric person, the person that sold actually curtains, that wanted to look at the fabrics really close under a microscope to know what was a really good quality fabric and what wasn't, which we actually have a lab on doing that too, said magnifying glass isn't enough. I need something more powerful. And what he did, and you can do this too. So go ahead and take a piece of cardstock out of your envelope. got one card like this. I'm going to take, put a little tiny hole. So you can use probe toothpicks, probably the best. Okay. I'm just going to put a little tiny hole here. We'll put it over here, actually in the center. Tiny. Make sure that piece of cloth doesn't come back. Right. We're going to take a little tiny piece of tape. Here. Same thing, we'll take the toothpick. Toothpick can score your tape. There we go. And now you can just rip it across. Boop, like that. Let's make this hole just a little bit bigger. Yeah. Okay, gonna make it a little bit bigger. What the heck? Let's make it a little bit bigger than that. So I'm gonna wobble it around a little. Okay. And then on the bottom side, I'm gonna put my clear tape. Just like this. Okay. Now, I have a water. Put a little bit of water in the bottom of this cup. And make sure it's clean. I'll take my eyedropper. Actually, let's do one more step. Let's go ahead and do one of these rings. I don't want to make a mess. So with these rings, normally when we use this on a slide, we call it a well, because it creates an isolated area that I can put something like water in, and it helps hold the water. So I just want one good drop there. Wow, that's like perfect. Okay, so can you see? One little drop of water on top of that. Now, that's so perfect, I almost don't even know if I needed the tape on the back. But now if I bring it down, you can do the same thing. I can look, magnify. So I can see just the roundness of that one drop of water magnifies the image down here. And if the hole is not big enough for you, do it again somewhere else. Make a bigger hole. Maybe make a hole the size of the paintbrush. Put the tape on the bottom, put the ring well around, and put a drop or two drops, just so you have that curved top. Because we'll talk about this later, but the curved top's really important. You can see it magnified. Some other things you can do is, if you have an extra light, bring a light over and shine it underneath to make your paper more bright. So bring it down, take a look. It magnifies. The original microscope was the same idea as this. In fact, there's a picture right here in your book. So this is Antoine van Leeuwenhoek. Actually took a little slit of like a brass, had a little tiny hole in the tip of it. And in that hole, he put a glass lens. He was really good at polishing glass lenses. Polished it really well, put it there where your water's at, put his specimen, drop of water, on the other side, and then would hold it up to a light, maybe a candle. The original picture I saw had him holding it up to the sun. Not a great idea, but the light would come through, amplify whatever's in that drop of water, and then magnify through the lens. And he could see little tiny things called animolecules. I need my animolecule picture here. He said they're molecular, tiny, microscopic animals that were living in this water. And that was really one of the first times that we had documented, or someone had documented, microbes. Later, as people played with it, thought, hey, this could be useful. Van Leeuwenhoek, who is actually, like I said, a seamstress, worked with fabrics and curtains, had started documenting the things he saw. He would spit on the end of it and see what was in his spit. He'd take scrapings of his teeth, scraping the inside of his mouth, blood, whatever he could find, droplets of water, he'd put on there. Just was fascinated about what was going on. we expanded that, and actually Robert Hooke had the same idea to look at like slices of trees. And when he looked at slices of trees, he found these little tiny cell patterns that we now call cells. But they were cell patterns because they were named after the monks' living quarters. If you looked at the monastery, the monks had cells that were stacked, I guess like a jail or whatever with your individual cells. But he noticed these little blocks that were in the foundation that held the tree together. And we'll actually do the same experiment that he did later. Other types of scopes, there are a lot of more powerful scopes than what you have as a compound light microscope. You can have stereoscopes with both eyes instead of monocular like you have. You can get electron microscopes that instead of using light, they use active electrons, and they can see a lot of little detail. You can see the three-dimensional detail. Oops, it says the cork image taken out. We'll put that back in. Okay, but here is actually a scanning electron microscope of the surface of a cork slice. Here's a little wad of cork, and there's actually one individual cork cell that he looked at. But there are a lot of fields. It can be, you know, if you're a botanist, which studies plants, that's perfect for you. If you're an anatomist that studies, you know, anatomy, human anatomy, it's perfect for you. If you're a geologist or doing forensics, or if you're a chemist, we're gonna explore some of those different things. All of these sciences use microscopes. In fact, almost every science I can think of uses microscopes in some way. Okay. So let's go ahead and talk about how vision works. So vision works because of light. Light sends signals, transport signals. It's like this visual telephone type of thing. I don't know what I'm saying. We have smartphones everywhere, but so when light travels typically as white light coming from the sun or other sources. So you've got all these different wavelengths that are together into this white light. But light works in funny ways. Light can be bounced. So let me see if I can get this to zoom on my book. All the way down. No, I might have to get this another camera to do this so that we can look at the book at the same time, right? So there are two important things about, or a couple important things about light you need to remember. One is all the colors that you know, red, blue, green, orange, all those are actually in white light. White light contains all those colors. Light can be reflected, which means to bounce off something like a mirror, or it can be refracted, which means to be split apart, right? So reflection, when you look at a tree, you probably learned this in grade school science, but when you look at a tree, that white light from the sun hits the tree, and the tree absorbs all of the colors except what? What's the one color it doesn't absorb? Green. It reflects, it bounces, whoop, that light off, and that light coming into your eyes tells you there's some object there that doesn't like green, right? But we see it as green, which is kind of cool. If you look at something that's white, that thing absorbs all of the light, and we just see this blankness, this whiteness about it. If it's black, black absorbs all of the colors. It doesn't reflect anything. It takes it all in. So black actually takes all that energy into the surface and absorbs all the colors. It's the absence of the colors. That's what black is, right? That's why when in the sun, you touch something that's white, it's not so hot. You touch something black sitting right next to it, it's very hot because white bounced that color off. Black absorbs or brought in. That's why in the summertime, you wear white clothes, you're cooler. You wear black clothes, you get hot and sweaty really fast, right? That's reflection. When we use a microscope and we use what we call top lighting, which is this light up here, by the way. I put it on wrong again. Oh, my batteries are dead in this one. Nuts. Okay. The top light up here bounces off your subject and then sends that light signal up your eyes. It reflects, right? Refraction is the other way. Refraction is when you take something and you send light through it and it splits the light. When we use the bottom light down here and we send light through, the specimen that was sitting right here, the specimen will split the light in different directions and distribute it. Okay? Refraction bounces the light or splits the light in different wavelengths. So like a prism actually refracts the white light and turns it into all of these different wavelengths, separate wavelengths, so you can see it. A lot of things do that. A lot of things do that and we don't even know it. Like we'll look at sand later. Sand looks plain. I mean, if you look at sand close up, sometimes it's clear, right? but sand actually bends the light and splits it in different directions. That's called refraction. So our underneath light coming up, a really good example of this, and you can try this. You have a cup. You've probably done this before. Oh boy, it's might be a little hard to do, but let's put a little water in our cup. Right? And if you take your pencil, put your pencil in at an angle. I don't know if I can see how it's shifted off a little bit, right? If I look up here, I'm sorry, up here, you see the pencil's going, whoops, like this, but then it's kind of disconnected at that point, almost like it's a different pencil right here, right? That's because of refraction. The properties of water, light doesn't go through water perfectly. It actually splits it and bounces it a little bit. That's refraction. Water, glass, plastic, lots of different things do refraction. They bend the light. If you ever looked at somebody that's in a pool and they look like they have little short legs, right? And their torso is really long coming out of the water, it's because of the light bouncing through there is split and it's refracted, right? So depending on what the material is, they have different refractive properties. Gels, waters, glass. Glass is amazing for refraction. Glass is great and we can use it to magnify with precision. Water will magnify. If you know, we saw that with that little droplet over there. It's the same thing with your cup. And if you hold it at different distances, sometimes it can change the magnification. With a flat surface on the top and a flat surface on the bottom, it's kind of hard. So another experiment you can actually do we'll do on the next page. Maybe I forgot it's on the next page, right? So it's refraction. A really important thing to remember is that, like I said, water refracts, plastic refracts, glass refracts, but so does air. If you look at a foggy day and you're looking in the distance, the reason you can't see things clearly in the distance is because the water is being refracted all the way through the fog, the little microscopic droplets. Same thing with rain, right? On a clear, dry day, you'll see the furthest because there's the air is not thick with water. It's very, very thin with water, so you can see very far on a dry day. Okay? It's the same idea of refraction. When we're looking through a microscope, there'll always be a little, with these microscopes, there'll always be a little bit of a distance between the end of this lens and your specimen. Air is going to change the way it looks during that short distance. So there's something you can do with microscopes, which you can't do with this one, but you can buy a special lens if you're interested in going in that direction. It's called oil immersion, where you put a drop of oil and you'll actually put the lens right into the oil to eliminate the air completely. It's the same idea with this. Oh, sure, now I can get it off. Okay. Let me show you. I have another cup here. Do this with. No, we do have a bunch of cups. So if I take these little gel balls, I love these things. Put some in there. I don't want to get the water in. I just want to put some of the gel in. You're making a really gross sound right now. If I take something like that, see my face, okay? And dry off my pencil. All right. Sorry. I'm just gonna write the word stop. Weird, why did I write it like that? That's strange. But if I actually take this and I put the word stop behind it, you can't see it, can you? Right? You can't see it. But if I take I have a lot of water bubbles in there. So let me do a little tap, 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 like on my computer. So it has some water bubbles, but if I take it and I put this up like this, you get a bigger glass for this experiment. Now I can see it, right? Because I've eliminated the air that was between those different gels. I definitely need another glass that has a better shape to it. Nuts. It didn't flip it. Okay. But now I can see the picture through. I'll have to fix that. And it's because I've eliminated the air. So air is your enemy. You always want to, you want to see something in really close detail, you want to get rid of the air, as much air as you can between it. And we'll look at that later. Okay, next. Your eye refracts light, just like a microscope. So if I look at my eyeball, it's like this. Let me switch views. That. Okay. I have an eyeball. It's the wrong eye color for me. Right? And we look at what's inside the eye. Light comes through the front, okay, where it's refracted by this thing called your cornea. The front of your eye, the white part of your eye, is actually continuous with this clear part called the cornea, okay, and it bends light. Your cornea actually bends a lot of light. We don't give it credit for it because it's hard to move your cornea. In fact, when we're older and our vision's not so well, like this, when we start squinting, what we're trying to do is we're trying to bend the cornea to manipulate the light coming through it, right? So the light goes through the cornea, then it goes through this little tiny hole called a pupil, which can be adjusted in size. And we'll look at the microscope, and you'll see the microscope actually has something like this called an iris diaphragm, which this is called your iris, right? To adjust the light that comes through it. Next, you have the lens. And this is what we give the credit for, because your lens can be manipulated by little tiny muscles that sit on each side over here. So if your lens gets really round and fat, you see things close up really well. If it gets really flat and stretched out, you're trying to see things far away really well. Those muscles can stretch and bend your lens to adjust how far the light goes. Right? This plastic's not that great, so it doesn't do a lot of magnification. Right? The next thing it goes through is not a hard substance like this, but this is actually a jelly. Okay, we call this the vitreous humor. So coming in the eye, you have the cornea. There's something called aqueous humor. It's a water right behind it. You have a lens, and you have more water. No air inside. It's all liquid. It's for the same idea, right? Think of those little jelly balls as the vitreous humor. It's a jelly. If you've done the dissection STEM lab with us, you saw that when we dissected the eye out, you had that blob of jelly that actually had the lens that became hard like a marble, and right behind the iris. Vitreous humor. Then the light comes all the way to the back, and finally hits your photoreceptors. The difference between your eye and a microscope is just the tools to amplify the image. Your eye is actually a visual receptor. It receives the image on the back. So the light has to hit all these receptors on the back. They don't go all the way up to the front. At least I've seen some debates on that, but these things are called cones and rods. Okay? When the light comes back here, they either focus on rods that help tell you black and white, so where there is light or where there isn't. So if you just saw on black and white images, or light and dark. And the other ones are called the cones, and I always think snow cones are colorful, right? So there's snow cones, and there's a little small tiny area of snow cone right here where you're trying to focus. Okay? If light goes there, you have very good color perception. If the light goes anywhere else, you have good black and white perception. So what's really interesting is that there are two things about this. One is that in your eyeball, that little area for color is only good during the day when there's a lot of color, a lot of white light. At night, it's not very good at all because there aren't many black and white receptors, the rods in there. So what's interesting is if you want to see something really clearly at night, like a sharpshooter, when they're aiming, right, like this at night, a sharpshooter is taught that you look at your subject and then you shift your eyes just slightly over to the side, because you don't want that thing at night hitting the color area of your eye. You want it to hit the black and white where you get a better black and white picture, and then they shoot as they fire. Okay? This little area called the fovea is packed with color cones. Okay? It's the primary place that you see color light. The rest, the black and whites, are all out here. So if I were to take this, if I didn't know what color it was, brought it from the side of my eye like this, over here's lateral. I can't even see it with my eye right now, but if I start bringing it around, you do this with a friend sometime. Bring it around, tell them keep looking straight, don't turn to the side. And as soon as they see your hand over here, tell them, say stop, stop, and then ask them what color you have. I actually have a little ball a hacky sack or juggling ball that I play with people with because it has 4 colors on it. And as I bring it closer, they don't know what color they're going to see and they have to guess it. It's hard to see because the light coming in here from the side is actually going in and hitting the opposite wall where it's only black and white being detected. But as soon as I bring this towards the front where it starts hitting the fovea, the light coming through hits the fovea, now I can tell what color it is. So fun games you can do. Maybe too much information. Might have to cut some of that out. Boom. Right. That's how your eye works. So for funsies, let's make a lens right now. Take one of the little Ziploc bags. And you were given some bigger Ziploc bags, but I will just use I'll use this one. It's kind of big. Right. You're gonna take some water. I'm gonna put water in the Ziploc bag. Well, I hope this doesn't have a leak. I haven't tested it. And it's actually a bag that was holding other bags, so we'll see. Boom. Just like this. Oh. That's why you want to have paper towels around all the time. Right? I'm going to squeeze it down. Here's the tricky part. Squeeze it, squeeze it, squeeze it. What do I not want in my lens? I don't want air. So I'm going to do this. I know this is clean water. I know this is a clean bag. And I'm just going to suck the air out. Okay. If I left a little bit of air in it, that's fine. What I'm going to do is I'm going to hold this up now. And I'm going to magnify with it. Perfect. Oops. I have a leak. I have a leak. Okay. So by taking this bag and put a little bit of pressure on it, I can make it nice and round. And as I look over my papers, oops, let's see if I can shift this around for you. This is exciting. This is working great. Let's do top. Yeah. Okay. I don't know if you're going to be able to tell, but see the words magnified? And if I flatten this out, I can still see the words through it. They're just not magnified anymore. And I make it nice and round again. I'm trying to magnify it for you. That. I can magnify it. So if you're ever in a pinch, you need a magnifying glass, grab a Ziploc bag and some water. Make sure your Ziploc bag isn't leaking like mine is. And you've got a handy dandy lens. But you notice when it's round, you actually see things close up. It magnifies. When it's flat, not so well. So when babies are born, they have very round lenses. Very round. What do they see? They see things close up really well, right? As we age, we don't have that stretch. So our lens gets flatter and flatter. What do we see better as we get older? Things far away, right? That's how magnification works. The shape of the lens, whether it's water or glass, plastic, determines how much magnification you'll get. Put that knot next to my other microscopes. I know it's going to leak everywhere. That's magnification. There are a lot of other factors you have to think of besides magnification, like resolution and contrast, which we'll go through later. Right, next, your microscope. Now we're going to break this thing down. We're going to look at one piece at a time to make sure you know your parts of your microscope. I've also put QR codes on the bottom so that you can do a virtual microscope. So if you want to play around and test things and see how they're supposed to work after this video, now if for some reason it didn't work right, you can. Okay, so as we go through this, hold this up. You can see this is actually a picture of your microscope here. So the first piece is starting at the top. First piece is called your ocular. Okay, your ocular or eyepiece or ocular lens eyepiece. Right now you have a cap in here. We'll pull that out. Have this sitting on the table. I'm going to put the tunnel chamber Barlow lens in here. We'll talk about how that works later. And then I'll put my first magnifier, my ocular piece in right here. Okay. So if I look through this, and you can do this too, it says 20x on the side. If I look down here, that is a good example. I'm getting that image amplified 20 times. Right? if the focus is right, and it's a lot of numbers and math that goes into it, but this will amplify or magnify, sorry, 20 times by itself. It's a glass lens, high polished, great shape, take care of it. It'll last you a long time, right? As I go down the tunnel, I go to the next piece. By the way, this will rotate. Oops, you have to loosen it up. This one's not loosened, but this will rotate so you can spin around and look at it from the other side if you want, depending on how you like to hold your microscope. This is called the revolving nose piece. Right? The revolving nose piece revolves. You can feel it click. If for some reason you're looking through here and you're not getting a good image, make sure that's clicked into place, because that's one of the first things that I would tell anybody that's right in person with me. So when you start, you always want to go to the shortest objective. So this revolving nose piece spins. The shortest objective lens is your red one. So this amplifies, if this were taken off, magnifies 4 times. So I have 20 times magnification here times another 4 times magnification. So I actually have 80 times magnification, and we'll do that explanation later too. So whatever I look at on this stage, which is the next part, is magnified 80 times when I look at it, right? So these are ocular eyepiece, lens, ocular lens. You'll have a 10x, you want to put that one in too. First, revolving nose piece, objective lenses on the revolving nose piece. I'll take it down like this. I don't know which is better, probably like that. Okay, objective lenses. Next, top illuminator, which is the light here, and we just did this earlier, but your illuminator button is back here. So number 1's top illumination. Central is off with a circle. Remember, the O on any power device is always off, since we're off, broken circuit. And 2 is your bottom illuminator. So illuminator is a fancy word for saying the light, right? So these are really strong LED lights. They work really well. Okay, next, you have your stage. These are called stage clips. I love it because cheaper microscopes usually have stage clips that are hard to work with. These are great because they're spring loaded, so you can just push them down, put your slide is the thing you're gonna look at underneath there. That little hole is called the diaphragm. So some microscopes will actually have an adjustable diaphragm, an iris diaphragm here. Sometimes they have a little bar, it's called a condenser. Yours don't have those, so they're not listed in here, but there are special pieces for other microscopes that you can add on. Right? Whoops, that's why you should always hold it with 2 hands and flat. You actually have a rotating diaphragm. The catch is your diaphragm is always set to a specific size. So as we go through, hold this, you can see there are different colors. The clear one right here is one you'll typically be using, so it's completely clear. Right? There's a little piece of glass in there. That's the condenser lens. So it will actually mildly focus the light to go right into the center of your specimen. That's the condenser lens. This will rotate so you have filters on there. And we'll get the filters later and how to use them. Right? Going down, and there are other attachments. You can see there are screw holes up here that you can get special pieces to do special functions or for safety features if you're really clumsy with your microscope, whatever you need to add. There are knobs on the side. So right here, remember this was called the arm. Just off the arm, you have a large knob. This is called your coarse adjustment. And if you turn it, you notice if you turn it one way, it lowers the stage, increases the distance between your lens. The other way raises it. Okay. When you're starting off, you'll always want to have it raised so it's closest to your lens when you start, because I can see to make sure that my lens is not going to poke through the glass. But when I have my eye here, I'm not paying attention to that. So if you start at the top and move down, it's a good thing to remember. And I'll keep saying that over and over again because I really want you to remember that. Coarse adjustment. Coarse adjustment's only used with the red lens. Right? And I'll show you how to go through these. The next one's a fine adjustment. Fine adjustment is used with all the others. The coarse is never used with the longer lenses, only with the short red one. Okay? Fine can be used with all three. And then the base is on the bottom. Okay? I turn it around. I have a rheostat. This just adjusts how bright it is. So sometimes you don't need full brightness, like bright, bright, bright light. Sometimes you just need a little bit of light, just enough. That's what the rheostat's for. Okay? And then I did mention on the back of the base, you have your illuminator switches, right? Power switches. And then of course on the bottom, you have your battery compartment. That's a brief overview of your microscope. We'll keep coming back to it over and over again. I'm going to put the cap back in for now because I'm just like that. I typically like to keep the lenses in a Ziploc bag, and I like to keep the cap on. Right? And when I'm not using this, I like to keep it covered too. But go ahead and give that a try. The QR codes. There's actually a virtual microscope lab on the right QR code, and then the actual virtual microscope on the left. It's kind of fun to play with if you're not playing with your actual microscope and you just want to get used to adjusting and focusing and things. Right? First look, this is fun. We're about to start using our microscope. Right? Okay, so are you right-handed or are you left-handed? You're left-handed, lefty. I know you're watching, Scout. So if you're left-handed, that doesn't necessarily mean you have a left dominant eye. You could have a right dominant eye. Handedness is actually not an ultimate either. So you're not just left-handed or just right-handed. You're actually varying degrees. You're more left or right, but if you notice, you do a lot of things left and right. Some people are very, very much split, and they'll do things like writing mostly with the right hand, but a lot of other things with their left, or vice versa. So your eyes are kind of that way too. You have one eye that does a better job of focusing and tracking than the other eye, and that's called your dominant eye, and that's not based on your handedness or your brainness if you're right or left brainer. Right, so here's what you do to find out what you are. So you're going to make a circle with your thumb and your first finger, right? So thumb and first finger just like this. Okay. You're going to look at something on the wall. Let's say I'm looking at you, right? So I'm looking at the camera right now with both of my eyes, okay? Looking at it. Now I close one eye. I can see that little camera hole right there. Okay. I'm gonna close one eye. I can still see the camera hole. I close the other eye and suddenly I can't. My right eye is open right now. So which eye was it for you? If it moved when you closed your left eye, then your left eye dominant. If it moved when you closed your right eye dominant, right? So I went like this, I closed my left eye and it jumped, right? My right eye is still open. My left eye is the dominant eye. That's the one that really tells my brain first what an object is. So when I close this and suddenly something shifts, I'm depending on my right eye, my weaker eye, left eye. And that's interesting too, because when I get prescriptions filled, it's my left eye that actually doesn't need as much work. My right eye needs a lot more work. So my brain was used to that. When do you think it would be useful to know which eye you are? I'm right-handed, but I'm left-eyed. When I'm playing darts, right? If I'm playing darts, like that, it's good to know. If I'm shooting a gun, it's good to know which eye I want to use, right? So it's not necessarily the one that's closest to the barrel. It might be the other one. If I am an archer, if I am throwing a baseball, if I'm doing whatever, it's good to know which eye is your dominant eye. Right? When I'm looking at something with detail, like if I'm looking in a microscope, I might use, or I actually usually like to use my dominant eye. But, so which eye are you? Figure it out. All right, setting up the microscope. Now we're going to go to the scope itself. I'm going to move this stuff over. I actually have one microscope set up. I'm going to move my samples over. Just like that. And I'll do the old cowboy switch. So I actually have one set up. I'll bring it over. Pressing it out. I will try and keep just like this. Okay. So usually you can see what I'm doing right here. You can see my table space, my tools. You basically see my lab in the situation. You can see me up here, and then you can see my specimen. So first thing you want to do, I'm going to take this off. Set to the side. Okay, so first thing I'm going to do is make sure that I have a level surface. This is nice, stay table. I'm not sitting on my leg or anything like that. It's a stable surface. Second, turn on the light. So I flipped on the light on the back. Right now, you see, if I tilt this, whoops, my bottom light's on, top light. So I want my bottom light on. I'm going to rotate the revolving nose piece so that my red lens, this is called the scanning objective. I'm going to keep using these terms over and over again so you're used to it. Scanning objective or the low power objective. So my red objective, remember, is the bottom lens. Up here is the ocular. I rotated the nose piece to make sure it's there. I made sure it was clicked into place, all good. I'm going to position my slide in the center of the stage. So this is my slide right here. I'm going to look at an angle. Here's a trick, and here's a nice thing for you to do until you're used to looking at slides. Okay. I'm going to take my marker. Right now I have a housefly leg. There's the actual leg. I can see it. Can you see it? Right there. I'm going to come down here to the very edge of the slide, and I'm going to put a mark. Just a marker mark right there on the edge. You can barely even see it. Marker back. So what I'm doing is I'm going to look for the edge of the slide, and then I'm going to find the leg. Right? So that's one way to do it. The other way to do it is I'm going to put my slide down, put my thumbs on spring clips. See my thumbs here? Push both those down with my thumbs, with my pointer fingers, index fingers. Slide these in. My clips over it. And on the side, I can see my specimen. And by looking at the side like this, I can center my fly leg right in the middle of that light. Right in the middle. So when I'm looking down here, hopefully I'm going to see it. But from here, I can definitely see that it's going right in the center of the light. Okay. My stage, before I look in here, I want to make sure my stage is all the way up, not down, up, so that my stage is as close as it can be to the objective at this point, right? That way when I turn it, I'm only getting safer by bringing the stage away from that, so I don't break the glass, right? Now, you're going to do this without this scope. You're just going to use your regular eye, right? So whichever eye you feel comfortable with, you're going to find your specimen. When I put that marker on the end, what I want to do first is I'm going to pull this back a little bit. I'm going to slide this down so my mark is right at the bottom of that light, right, centered. And I'm going to start looking through here and watch for the edge of the slide. You can see it. Anywhere that that stage is at, you're going to see kind of a real blurry line coming through. So I'm going to hook this up so you can see it. I'll show you how to hook this up later. You have the same camera. Look how blurry that is. Crazy. See that? All blurry, blurry. All right. Remember, it's all the way up right now. I'm looking through there, looking. And what's nice is even if you just have the camera, it's not hooked into a computer like this, you can see this big image here. But we're going to use your regular eyes for now. So you're looking. Okay. Kind of clear. I'm all the way with my stage at the top level. I'm going to push down spring clips with my thumb, make sure my slide's underneath there. Okay, sliding into the light. Keep watching my microscope. Watching. So you're going to watch that image over there too. I'm bringing it down, bringing it down. Oh, see that? There's the edge of the slide. See it? Okay. I don't know if I see anything special. So I'm going to put that edge of that slide right there. Now I'm going to use my fine adjustment because it's pretty clear. If it's real blurry, I can use my coarse, but see, there it went. Straight through. So I'm going to use my fine adjustment, real slow, until I get a real clean picture. Look at that. See the ink? So if you look right up here, see that purpley blue ink? See how clear it is? Awesome. All right, here's where it gets tricky. And we're gonna keep practicing this over and over again, and later we're actually gonna learn the skills to be able to move this better, but you're gonna have to kind of guesstimate it first. This is like playing with an Etch-a-Sketch. I know you played with them. They're fun to play with, but they're kind of tough in the beginning. So I'm moving my slide around slowly. I don't want to go fast because I don't want to lose that little blue purple edge. There it is. Look at all of that. Are you noticing the way that you move it? How it doesn't seem to go right where you want it to? I'm telling you, it's like an Etch-a-Sketch. So there we go. I've got the whole blue image in focus. I'm going to try and level it out a little bit. Slowly. At first I went to turn the wrong way. Now I got to get back in the center. Awesome. That's pretty level. And now that I have it pretty level, in my scope, you can see, and if I twist this, I might lose it, but you can see my slide is actually really square on my stage. Stage is the black thing you put it on. It's where your actors work, your specimen's your actor. The slide is what you slide up and down. Now, remember, my marker is on the bottom edge of the slide. That's actually the crack of the glass going right across here, right? I'm going to pull down slowly. I want to pull straight down. I don't want to go around anywhere else because remember, my fly's leg is straight above that marker mark. Oh, what do I see? Cool. Now I see another edge of glass, and that's where my slide cover is. That's the thing that covers up the specimen to hold it in place. Look at that. I can see a lot of little air bubbles trapped in the preservative. So we'll use nail polish later as this preservative. Keep pulling down, keep pulling down. Are you? There it is. Perfect. Patience, patience, patience. And now I've got my fly leg right where I want it. Awesome. Okay. So I forgot to tell you, but your fly leg is in this little box. Now we'll probably have to remember to go back and do that again. If you open that up, it's one of the very first ones. And it doesn't even have to be the fly leg that you start with. It can be an onion. It can be anything. The tree. But these are the basic training slides for your microscope. Now, if I see the leg, I can focus in. Look at that. Blurry, blurry. I turn, and I'm using the fine. Blurry. I'm going to switch over so that you can see the details. There we are. Nice. Look at that. Cool. This is almost like a crab leg, you know what I'm talking about? I don't know if you can see my little marker hand, but up here, oops, down there, you see at the top of it, it has like when you take a crab leg and you break it open and you pull the meat out, you can see the shell. So the exoskeleton on the fly that goes down like at a V. I gotta get used to doing this backwards. Down like at a V, like this. You can see the muscle that's kind of hanging out, which would be the meat. You can see those little tiny hairs over there, just like your skin. Pretty sweet. If you go back and forth, we can take it in detail. You can actually see the knee joint down the bottom, tip of the V. Fun. So we kind of jumped ahead and did that. So you adjusted the coarse knob, you recentered your specimen, moved it around. Fine adjustment knob, adjust the diaphragm if you need to, which is the light underneath, which Our scope only has one unless you want to flip the filters. And then you can change the objective like this. So now that I have it, what I want to do is kind of center it. I want to see some of those hairs better. So I'm going to put the hairs right there in the middle, close to the middle. And then I'm going to take this, look down here in the bottom right corner, the revolving nose piece. You can feel it has kind of a grip there. I'm going to take it, and I don't want the longest one. So what's the longest one? Longest one is the 40X over here on the blue. I want the yellow. So I'm going to go from 4 to 10, then to 40. What this is doing is going from 4 times objective magnification to 10 times. Rotate it around, click it, blurry. Okay. So some microscopes are called parfocal, and they're very expensive microscopes where the center will stay lined up. But I found a microscope for you that's really close to being parfocal. So what you were focusing on in the center is really close to being focused here. Okay, or parcentral location. Sorry, parfocus means focus. This is just slightly out of focus. I can still see the leg. So all I do is the fine knob, the little knob on the side, not the big one. Fine tune it. Go one direction. It's getting clearer. If I wasn't going the right direction, I just go back the other way. Oh, that made it blurrier. Clearer. Oh yeah, look at that. I can actually see all those little tiny holes in the fly leg. I can see those hairs. So I'm going to try and slide it over real slow. Because remember, now it's not 10 times magnified. It's actually 100 times magnified because I have 10 up here for my ocular, my eyeball, times another 10. So 10 times 10 is 100. So I'm going to slide this just a little bit. So any little tiny movements are 100 times magnified. There we go. Nice, right in the middle. Okay, rotate this. Boom. So I rotated it one more time. I went from the 4X down here. Remember, I'm pointing right down here on the right, lower right. Move this over for you. So I went from the 4X, the red objective, to the 10X, which is the yellow, and now I'm at the 40X, which is the blue. So I have 10 times multiplication up here, 40 times here. So I'm magnified right now at 400 times. And we'll explain all that later. Look at those hairs. I can actually see the details of the hair. That's amazing. That side hair, this hair over off the leg is a pretty good one. But I'm looking at those pores. Those pores are pretty cool. You can almost see a real fine fuzz on the leg. So kind of like you have a little bit of baby fuzz on your cheeks, but if you're an adult male, you've got whiskers coming out. So those hairs are like the whiskers, and that little fine fuzz is like the Velux fur that mammals have on us. We all have fur, male, female, baby, old person, a real thin layer. So I'm just kind of real slow creeping along, looking at different things. Pretty crazy. And if you're making a lot of mistakes right now, good. I'm glad you are. Wow, look at that. You can see the layers of the hair. Right? So you can see the center cortex. If I look at the hair that's over here on the right, see how it comes out of the body. Oops. It's kind of thick. So it's thinning out. There's a black line right down the center. That's the cortex of the hair. And we'll look at hairs later. We'll look at a lot of cool stuff. But we'll look at hairs and talk about them in detail. I can see the cracks in the exoskeleton. So I can see the darker tan areas, and I can see where it's kind of cracked, where it's a light tan. See it? Very cool. Okay. So, whoops. While I'm on this setting up, I'll move this over. I'll show you the actual setup for the camera. All right, flip back over to my desktop. A little bit messy. All my tools are here. That's okay. As long as it's safe. Messy labs are okay. Unsafe labs are not okay. So when I'm looking at this, I'm going to slide this further again. Shoop. You can see this is an old broken cell phone that I had. And I'll set from scratch again. And to set this up on your computer is one thing. You can do that if you want, but really for you, if you have a decent size screen, you don't need to hook it up to your computer. I'm doing this for your benefit. I normally wouldn't hook it up to my computer because I can take pictures from my phone and I can just send the pictures to my computer if I want to do something with it, like if I want to make a PowerPoint or whatever. So here are the different layers of my setup. Let's see if I can get this back in focus. I know I'm only holding with one hand, but I've got a good grip here right now. Barlow lens went in. I have my 10X, right? This will come apart. Don't take it apart until the time that I tell you and walk through it with you, because we're actually going to put something special inside of there to help measure, but don't do it now. So I have a Barlow lens. I have my 10X magnification, the big optical lens. Okay, I bring it down. Okay. I've already done everything with my eye to focus. This is really important. I've done everything I can to focus this, and I have a really good image. In fact, you know what I'm going to do? I'm going to go back to my scanning, which is the red. I'm going to look in, make sure, yep, the leg is still there. I have excellent focus. Okay, I know you can't see that right now, but here is your phone mount. Bring this forward a little bit. This cap just goes right on here. You have to have one of the lenses that came with it. If you ever order different lenses, you have to measure to make sure you get the right one, but it goes right on here. Okay. If you want, if it wobbles too much for you, you don't like that it wobbles a little bit, and you can stick a little bit of paper in there, but it's not necessary. Let the phone holder hang. It's loose right now, right? Make sure that's loose. I'll take my phone. I turned on camera. Right? I've already turned it on, so I just turn on the camera. It's my screensaver. And normally I would see, you know, everything my camera sees, but it's projecting. Right? So it's actually right over here on your screen. All right, this is where it gets tricky. You might have an adult help you. I'm gonna wiggle it in, go like this. So it's holding on one side of the holder. I'm gonna pull the other side out. I'm sure you've done this before when you've put your phone into a mount, whether it's, you know, in a car if you're driving age, or it's not a desk holder. Right? So I have it on here. I'm not getting a really good image. See? It's because I have to make sure that the lens on the phone lines up with the lens. So I'm looking for circles of light over here. Kind of gently bring my phone down. This is where parents really could. Okay. Oops. I already fixed mine into place. You can loosen yours up. See how it goes back and forth. Right now I can see, whoop, there's on the side of the microscope. I'm just turning it, bringing it back up. I'm holding the adjustment on the back. I just tightened it a little bit. It's not locked down, so I can still pivot, but I want to be able to put it so that I can see a nice big circle. Oh, there it went. I just missed it. Here it is again. I missed it again. There's my circle. Okay, so I'm right about where I want it. Keep just missing it. Okay, snug this down just a little bit. I tighten this piece in the back. And trust me, you only have to set it up one time typically, and you can leave it there for all of your experiments. Right, I'm gonna put it at an angle. So my phone doesn't have to be perfectly flat in the mount. I can put it at a little bit of an angle, if that's what my phone needs. And I'm going back. Oh man, did you see that? I was right there. Tightening. That's a really good spot. It's nice and tight. I can see most of it. I'm gonna mess with it just a little bit and probably mess everything up, but oh, a little to the wrong side. Now I know. Now I'm just gonna adjust the top a little bit, little bit. Boom. Oh. Okay. So guitar string. You slightly overtune, focuses it. It comes back in tune. Look at that. Is it perfectly focused? Nope. One thing you might want to do is turn off the autofocus on your camera first, your phone. But now I don't have to mess with the focus on my phone. I leave it alone and I can do fine focus, right? So now I'll switch back and you can see what I'm doing from the scope. There it is. So see that? Oop, a little dirt. The slide's not perfect. Looks like it has some dirt on it. The preservatives kind of had a lot of air in there. But yeah, I can see, totally see. So if I want, now I can do the same thing. I can turn my revolving nose piece from my 4x, red 4x, to my yellow 10x, a little off center. There it is, pretty. Can you believe that is a fly's leg? That's insane. I'm gonna do it one more time. Whoa, look at all that detail. Crazy. And then they're back to the hair. You can see the cortex in the hair and everything again. That's how you set up the mount. If you don't want to set up the mount to do the camera and you just want to skip that section, that's fine. If you're having problems or struggling with it, that's fine. If you ever want to come in, if you're in the area and I'm at the lab, I'll definitely help you. So don't be afraid to reach out. If I'm here, I'm definitely willing to help because I love playing with this stuff. Okay, that's your setup. I'm going to move this back out of the way. 2 hands on my scope. Shift it to the side. I'm going to flip back over to the book. All right. So I put in here first looks, but we've actually taken a pretty big first look. And I might have to consider not doing that. Maybe, I'm not sure. There's the fly's leg. We already looked at it. So why not look at your parts of view? So sometimes you don't need a slide. We can take that slide right out. If I'm going to use it later, I can put it on a piece of lens paper. But I'm opening it up. I'm making sure it's going to slide in here safely. Click. Went in both sides. Be safe. Click. I didn't have to force it. Perfect. Okay. I'm going to drop my stage all the way back down. Okay. I'm going to flip to my scanning lens. Right. And then I'm going to try the top light. So all the way here in the back, on the bottom of my base, remember, number 1 is the top. Now let's go from the top. I'll put my finger in there. Actually, let's look at the bottom. Sweet, look at that. It's my fingerprint. You see all of the little epidermal ridges. I'm going to move this clip out of the way. Just put my finger right here and hold it. That's pretty sweet. Yeah. So you can do that. I'm going to flip my finger over and look at the top of it. No ridges. Just old skin. Slide down. I want to see if I can see my fingernail. Huh. I have a scar right there. Nice. There's my fingernail. See if I can focus in on any of this. My finger's too fat. Oh, there it was. Missed it. So make sure you're not pushing down on the stage. You don't want to be pushing on it. Yeah, see there? There's the surface of my fingernail. There's the cuticle. Ah, that's the edge of the fingernail. Gross. That's really gross. But that's where here's my skin. Here's my nail. All fingernails are special appendages. Like we talk about your arms and legs being your appendage, things that hang off your body. Your skin is like in the picture. It's an actual organ. And your nails are appendages or extra organs that hang off your skin. So here you can see there's my skin. There's the edge of the nail, the cuticle. Boop. Oh, gross. You can see the layers. And then there's my fingernail nail. I smashed my nail. I have a white streak in it. Oh, you can see it. Sweet. Let's go all the way in. Let's see how bad I need to trim my nails. Ooh, what's that? Do you see that? What have I been playing with? Crazy. Is it bacteria? No. It's a piece of moss, I bet. Insane. I'm going to see. I don't know if this will work because my finger's pretty fat, but I'm going to try and go to a higher objective. Oh, just made it. There it is. Oh, I missed it. There it is. That is totally a piece of moss. Or some microscopic green caterpillar. Which, by the way, if you didn't know, you've got microscopic little mites growing in your face. We're going to find some of those later in one of the other modules. So I am going to take a toothpick if I can. Oh no, I'm going to lose it. Well, I won't be able to. Time's sake. Okay, so I just slid everything. And I lost it. But I could take a piece of the toothpick and scrape that thing off and maybe put it on a slide and check it out later, which is something I totally would do if I had more time. All right, so look at your skin, look around, compare the back of your finger to the front of your finger, look at your fingertips, your actual fingerprints, look at your fingernail, check them all out, right? I would say look at your arm if you could, but you can't get your arm under there. But if you have a hair, and this is tough, maybe you can pluck a hair out and just put it on a piece of plastic and slide it under. There are a lot of cool things you can do though. All right. So when you're doing this, always take notes. So for sake of time, I'm not writing a lot down, but what did I observe? I observed my fingerprint. And what do they look like? Ridges. They almost look like little hills and valleys, like a hill and valley. I could see what looked like scales almost, so it was kind of scaly. I don't know if I spelled the word scaly right, but just documenting. So if I were to draw it, I saw this round image, and I could see there was a ridge here. It was kind of deep going down. There was another ridge here, kind of deep going down. But just doodle, draw what you think you saw, right? When I flipped it over, I saw my fingernail. I saw the cuticle edge that was kind of rough here. I could see kind of a weird rough edge, and I could see skin cells again. So I knew this was skin. Oops, wrote right on it. I could tell this was the nail. I saw that weird white crack, right? And it was kind of like a rough edge, almost like something folded over here. And then if you didn't notice, there was kind of a like ridge right along here, almost like it was another nail, but real super thin. So just the things you notice, jot them down, put them down, and then look it up later. If you didn't know this, Google, it's spelled Google, knows everything. So if you're wondering about what you saw in this picture, just Google fingernail anatomy. It's fingernail anatomy. And don't be afraid to write this in your notebook too. This is what I need to get Google. And then go back and label it. What did I see? Okay. Did I see the hypochondrium? Did I see the nail bed? Did I see the cuticles? Did I see skin? What are the things that you saw, right? So here's a little note. So what did you see? Describe it. Use words. If you need to describe a smell or whatever, do that. What strange things didn't you expect? Well, maybe you didn't expect to see that fold right there along the fingernail. How clear could you get the image? What was your resolution? We'll talk about that later when we go into the second section. So there's a difference between magnifying something or making something bigger and actually resolving it to actually get clarity of the different parts. We had pretty good resolution. We could see the lines right along here. That's awesome. Did we get the image we wanted? Can we go to a higher objective? We did, sure. We wrote down the objective. We looked at this at 4x and 10x, right? So 4x was our objective times 10x, which was our ocular. So what's that? 40, right? It's 40 times magnified. We call that the TM, or the total magnification. We'll do that. We'll spend a whole section talking about that. So these little things I keep throwing out, right now you're just playing. The whole purpose of 101 is to play, get your hands on it, do stupid things, make mistakes without breaking anything, safely. Okay. How big do you think these things are in millimeters? Wow. Geez, my finger's probably, I'd say 1.5 centimeters. So this whole thing here was probably maybe, gosh, maybe a 0.5 centimeter. I'm guesstimating. We'll learn how to measure later. This is called our field of view, all the things we can see. Did things move the way you expected? When I shifted my slide to the left, did the image go to the left? What happened? It's weird. Let's see, things didn't move the way you wanted. If you moved up, did it move up in the image? Where did it go? Remember how messy you worked and how hard it was to find things? Yeah, you're sloppy. It's okay. You made a lot of mistakes. You're going to hone the skills. You're just playing around right now. Don't get too hard on yourself. Practice, practice, practice. Right? The next thing we'll do is we'll analyze some pictures. So we'll just take a peek at some of these pictures here. I'm going to take this out. Your book actually has it so that these pages are like this, which is great. But I'm going to look at things. So I'm curious, if I looked at my skin, I saw that detail. What can I see like on this page? What about pictures? So let's try it. Let's give it a shot. I'm going to reset everything. Remember, when you're starting with something new, slide clips are in the right place. I didn't change diaphragm. Light's still there. I need to make sure my scanning, see the red line? Scanning is down, right? I'm the closest I can get. That's really close, right? So my stage is as close as I possibly can get to my objective. I'm trying not to move the microscope right now because of the camera that's on it. Moves back. Okay. As close as I can get. Now, we'll look at some things. So, observation. Pictures look different on different types of paper. I've noticed that. This maybe doesn't have the clarity that a photograph does, or it doesn't have the clarity that your stem flyer or your stem, excuse me, your Steam bookmark has. So it's shinier, it's glossier. This is not so glossy. The lines seem a little more crisp on this, not so much on this. So I just want to look to do it. Compare some different pictures. I'm going to take a tiger because I'm really curious about this tiger. I'm just going to slide a tiger in here. Same way I did before. Push the spring clip down with my thumb. Slide the tiger back. Oh yeah. Ooh. Cool. There's the edge of the paper. Well, now I could actually focus. That's what I'm going to do. Focus in. Look at the paper. It's pretty sweet. I can see the roughness of the paper. Slide it back. Slide it back. I'm going to take a peek at that tiger. What color is the tiger? Yeah. Yellow, green, and red. And blue, right? Not so much. That can't be right. Got it again. So I'm looking right next to this hole. That's all orange and black, right? Let's check it out again. I'm going to slide it down. I'm going to take that spring clip, lift it up. I'm going to go right next to the hole. In fact, right now, there's that hole. That's kind of cool. There's the hole. There's the edge of the paper where it got punched. You can see the roughness. It's like little hairs, right? So I want the other side of the hole. So I'm right here. Whoops. There it is. There's the edge of the hole. Oh, wait a minute. Well, that's not right either. So I know there's black on the tiger, but weird. Let's see if I can focus in. I'm going to go ahead and switch this back. What? Huh. So I see some yellows, reds, and blues, and every once in a while where a blue overlaps with the yellow, I see a green. That's got to be a problem. Oh, still top lit. I'm going to go a little bit closer and see what happens. Here you can see the orange hidden back there. All right, I'm going to refocus. Fine tune knobs, the little tiny knob here. Oh yeah, nope, no orange. Maybe some overlap there where the red is stuck to the yellow, but yeah, no orange. I'm still seeing yellows, reds, and blues. Huh. Let's see if I can find a stripe. Let's see what happens when I find, oops. let's go ahead and flip back to the scanning. I'm going to scan around. Anytime I'm moving a lot, I go back to that short lens, the red one. Down here, boop. Refocus, boom, whoa. Yeah, still no orange, crazy. Let's see what happens when I get to the black. Right about there should be a black stripe. Well, there we go. There's some definite black stripes. Well, the blacks are black, but that's the tiger's chest. There's a lot of white with a little bit of orange specks. Crazy. So what's happening? The colors you see aren't actually the colors that are there. So when you look at a printer, printer prints in typically 4 colors. You have red, blue, and yellow. Oops, sorry. Red, blues, greens, and then black. So you definitely see the black printed. Look at all those red, blues, and oh, there we go. So there's an art method that uses, it's called pointillism, where you don't actually see a whole color of orange or a whole color of green or whatever. You see little dots that add up to that color from a distance. And I actually put a QR code down here on the bottom of the page that gives you an example. There's a really famous picture, it's called a hybrid picture, where it's Marilyn Monroe and Albert Einstein. And when you get close up to the picture, it looks like Albert Einstein, but as you zoom back out, it turns into Marilyn Monroe, because they're manipulating your eye in the way that your eye takes images and blurs it together. The same thing with paper. When you're printing pictures, color pictures, they manipulate those little tiny dots, just like in this. So this whole painting, a series of little dots. And if you got really close, you could see them. But from a distance, it looks like an image. It looks like a really pretty image, but it's only little dots. So let's take a peek at purple, see what happens. There's the paper edge again. There was a hole, and my butterfly was right on the, oh, that's sweet. I got to take a second on that one. So this is the thing about microscopes. You think you're looking for one thing, and sometimes you see something really cool. Look at the fibers. It's amazing on that paper, but ripped. We actually have a whole segment module on fabrics that we'll do later, textiles. And I'll also have some pieces of United States currency and some counterfeit currency that you'll take a picture of too. Look at that. And we'll be able to compare and see what these things are made out of. What's a dollar bill made out of? What's a $100 bill made out of? Whatever. What's your paper made out of? Okay, so let's go around the hole. We're going to find that purple butterfly. Ooh, man, that looks kind of purple even from here. That's cool. But what happens when I get closer? So I'm focused. Boop. So right now my microscope's tricked. This thing is all purple too, but it's not. See? Those are reds and blues, lots of reds and blues. And of course, red and blue together make purple. But if I blur the image, well, now it's turning kind of blue. There it goes, purple. Mostly blues, pink, sweet. All right, I'm gonna pull the paper out. So anytime we pull slides out, we wanna flip back to our scanning objective, the red line. Pull this out. Let's compare it to something that's like a printed image that's on real nice paper, right? So we'll do that. Whoa. Wow, look at that. A lot different. All right, so I'm going up. Should get white, yep. So now I'm a white area. And now I'm at the robot. There's the red on the edge. There's part of his black face. There's his pupil, right? Sorry, iris, the color part of their eye. That's kind of cool. And I'll pull this out. And you see again, I was coming in here. I hit to the white background, saw the red edge, oops, on Rube's head, went into the black area, then went right into the eye and looked at the pupil. try different things. It doesn't have to be just this paper. You can take magazine articles, right? You can take real photographs. What's really cool is if you have like a, they don't make these anymore unless you're a professional photographer and you're using a non-digital camera, but color negatives. So if you take one of those little negative strips that came with old pictures when you get them at a photo shop or whatever, and take a peek at them and see, what do you see there? What if it's just a black and white picture, like a newspaper? Well, coincidentally, got that for you. So you have a small clip of picture. Take this little section. So right here, you can see there's a woman's face down the bottom, with a bunch of books behind her. Oh, I had tools, sorry, not books. So bring our stage up to the top. I'm just gonna slide this in. Lots of examples of not even needing a microscope slide to check things out. Right, I'm gonna put her in so that her face is right there. See if I can focus in on some of these. There's the paper. Cool. I'm gonna take the slide spring clips, pull them off. And I'm actually scanning over her face right now. So you can see there are a lot of dots, but then there's a lot of solid colors that you really didn't see on the other paper. It's because the type of paper makes a difference on how they print it, right? Because ink bleeds, ink spreads. You can even give that a shot by taking your marker, right? You take your marker, just, there's my marker tip, right there. There it is, there's the tip. Now I'm just going to, look at that. You see the ink spread? So wheat. Let's see if we can focus in on that spread. Watch it. That's pretty cool. Okay. But try that with different papers. Try just looking at the edges of the papers. Look at the edge of this newspaper paper. Newspaper paper. Look at the fabric. I mean, it already looks different. Thing we saw with the printed paper. You have a paper towel. You have that in your kit. I'm just going to rip the edge off the paper towel, like this, and feed it in. What do you notice about it? What do you notice is different? It's more porous, a lot more gaps in between the threads. And the reason that it's like that is why? Why do you think there are a lot more gaps or airspace in between the threads on a paper towel than there is a piece of paper? Because it's meant to absorb, right? So it's going to soak things up. So always keep your mind open and think of things like that. Just be inquisitive. I love it. If I have things around me, I just want to put things under the microscope. So let's go on to the next section. All right, navigating the specimen. Now we're going to get into the real skills of making things happen. So we're going to go for something called hunting the E. So you've noticed already that when you put something in there under the scope and you slide it around, it doesn't always go where you want. You know, if you slide it one way, it's like, wait, it's going the other direction. It's like an Etch-a-Sketch. It drives me crazy. But we're going to do a quick, easy slide where we look at the letter E. So you're going to grab your newspaper clipping. Or another one, whatever you have. Right? And then you're going to follow this procedure. When we put the book together, we put it so that you have a lot of information in the beginning of the lessons, but as we go further in the lessons, there's less information on how to do things and more information on what to look for. that makes sense. So I'll give you a topic, more information on the topic, and you should already have the skills to be able to decide what kind of slide am I going to use, or how am I going to look at this? What objective will I use? All these fancy words, you'll know those skills, just like learning an instrument, right? So we already ripped it 2 to 3 inch square section out of printed newspaper or magazine. Okay, you can even use the bottom corner of the book if you want, if it has a letter on it. Right? You're going to have a pen and a fine tip marker. I don't need all of that. And it says, follow procedure for setting up your microscope. Already did that. Flat surface. Now, if you want to come back and work on it from scratch. Okay. Start scanning power further from the stage. We did it. Without a slide, put the newspaper under the scope and center over the light until you see the letter E. Right? Let's try different lighting. Let's see what it does below. Oh, sweet. So there's under lighting, bottom lighting. We're going to look for a letter E in there. So I'm looking. Right now I'm seeing some are working for. Oh, actually, down here on the bottom, there's a letter E in distributed. So what I want to do, I'm going to slide it in here. The letter E is upright. It's perfect. Slide it in. Looking for some words. I'm going to use the spring clip. Look, I already found E. Sweet. So now I'm just going to try and get that E in there. It's a sketch, I'm telling you. Oh, there it is. There's the E. Whoop, went too far. Come back, come back. Okay. Well, let's try top lighting. Is it helping me any? There we go. So I'm top lit. There's a U for distributed. So I want to keep on UTE right there. It's tricky, very tricky. Whoa, too far. There was the letter D. Let's get that thing as close as you can to the center. Oh, I lost it completely. There it is. Oh, yike, yike, yike. Perfect. Okay, got it where I want it. I'm going to take my thumb, put it on the spring clip. I'm going to hold it down right there. I'm going to take the other one, bring it to the other side. I don't want this moving because I want to look at it a little bit. All right, that's the letter E. What's wrong? In your notebook, here's your experiment. You did, what did we observe? So we started this with an observation. Things don't move the way. So here's our observation. Things don't move as predicted. Let's say that. Under the scope. They don't. It's weird the way they move. It's crazy almost, right? That's my observation. Let's go back here. Excuse me. Cough. Taking this page out temporarily. Boom. Second thing, what's my question? Open your mind. What if? So what's my question? What if I looked at something I'm familiar with? If I look at letters, will they flip? Flip? And or move as I think, or move opposite direction, we'll say. Opposite directions. I mean, technically, that's 2 questions, but so my hypothesis is, number one, we noticed that looking through lenses earlier is that the microscope will flip, or the fancy word is invert, a letter. We're being specific though, it's the letter E. Flip or invert the letter E. Okay. And I'm actually, like I said, doing 2 questions here. And then as I move, and then movement, I think fast like right, is opposite in the scope. So I'm going to draw what I see. First, I'm going to draw a circle here. Okay. I'm going to draw what I see in the scope. Remember, I saw with my naked eye the letter E and distributed, right? What's it look like in the scope? Here's the letter E, the way it's supposed to be. So I'm seeing the letter E. Let's draw it like this, kind of like a big C, and put the bar across. Whoa, totally not right. So it flipped it, it inverted it, or mirror imaged it. Actually, mirror image is the proper term. Okay. So it flipped it over, creating a mirror image. And that's the first part. Oh, my experiment. We just did this letter E. Sorry. So experiment. Newspaper. Newspaper. So I want to focus on this. I'm actually going to look at the bottom lighting again. That's pretty cool. I can kind of see through the paper. It's almost ghostly. Look at the detail. I mean, technically, if I looked at some of the detail and I wanted to spend time drawing, my little E would have lots of little fabric patterns. It's crisp and clean on the newspaper, but it's kind of a little bit rough here. Right? Since I have everything centered, I'm going to rotate my objective. So revolving nose piece, flip it around. I went to the yellow, which is 10x. Right? So this first one was 4 times 10. So it's 40 times. 4 from the objective, 10 from the ocular. Let's focus again. Whoa, that's a big jump. All right, well, let's see if I slide that. I don't know what I'm looking at. I see the top edge there. So I mean, technically, if I'm looking at it, I've got a circle, so I'll draw my second one. And I just have, it's kind of splotchy across the top and then filled in with fabrics, right? And then down here at the bottom, see a little bit of the E, kind of, but it's the same thing, splotchy. So if I'm looking at this, it's a straight line on the top. This looks like a little bit of a curve with the circle. So I'm guessing if this is a straight line, I am looking right there, right? I'm looking right here. There's the bottom that's blown up right there. It's top right here. So this is actually like that. And that said 10 on the objective, and I already had 10 on my ocular. So that's 100 times magnified. I'm going to go one more step. I don't know what I'll see. The problem is if I zoom in the middle of this paper, all I'm going to see is more paper. I'm not going to see any print. So let's do this. I'm going to pull it down. Oh, spring clips are in the way. So I'm going to gently put my thumbs on the spring clips, pull them up, pull my paper down real slow. Oh no, I went out of focus. Oh, this is tricky. There we go. Well, wait a minute, I'm pulling the paper down and it's actually going up. Something's not right. Oh, now I lost it. So let's go back over to my E. I'll do that again. Accidents happen. There's my E. Swoop. Oh yeah, it's sliding away from me. There's my E. So as I slide the paper to my right, the image is actually going to the left. As I push down, it's like a game. Just wait, you'll get really good at this. So good that when you're chasing like or living things, like in pond water or this moss be able to catch them. Huh. Yeah, I did it again. That's not my other E, my original E. Oop. But once I get it there, get it here, focus, focus, boop, focus, turn my objective. You did already. Refocus. There it is. Whoa. There's the straight line, so that's in the middle. Now, I'm lucky. That's pretty much in the middle. I'm going to do one more. This is going to be a tough one. Oh. There it is. That's all I can see. That little tiny bit. Crazy. So hard to get it focused too. It's almost like glass. I'm seeing the paper so thin. I'm gonna play with fire here. Oh, there we go. Slid up just a little bit. Same thing. Look at that. Now I'm seeing that the paper isn't even completely stained. It's not even completely dyed, I mean. So I can see like that straight line that's now crooked. You can see a lot of roughness. I can see kind of another liney pattern here. That's all part of the straight line. So if I come straight down here, and that's at 40x times 10x equals 400 times. Magnified 400 times. So hard to get a good quick picture on that. And that's top lighting. It's not very good. Let's see what happens when I do bottom lighting. No lighting. Blow out. Whoa. Now the paper looks like glass. Huh. Isn't that fun? I can see the different layers of the paper. All those fibers I saw before. I can see some of the fibers on the top. Crazy. So get used to it. When you're moving in one direction, it's actually going in the opposite direction in your image. Know that things are going to be flipped backwards. So it's like reading a mirror when you're doing this. As you get higher power magnification, things get, wow, so much detail that you're not going to be able to see everything in detail. And we'll talk about that too. There are actually depths to everything you're going to look at, whether it's a cell or it's a piece of paper. And when you're focusing on something at really high power magnification, you can only focus on little layers of that. So if I have my fist, I can focus on the top, but I can't really see the stuff on the bottom. As I focus on the second finger, this is out of focus and everything below is out of focus, but just that one spot. And that's what I'm seeing as I'm going through here. I can see the very surface of the paper, so I'm not focused, not focused. Whoop, there it went so fast. There's the surface of the paper, some of the deeper fibers, oh, and then I'm through focus. So keep in mind, you always want to record notes. What did I notice? At the very end, my data, I collected, I analyzed, I drew pictures, and I noticed that it did mirror image. So here's my data. You can put charts or whatever. It mirrored my image. Excuse me. Edges blurred with more magnification, or when they're more magnified. I noticed that. Things moved in the opposite direction. Things moved opposite direction. Anything else you noticed? I saw layers. I could tell, I could distinguish layers. I saw fibers. I saw individual fibers in the paper. I saw individual layers as I'm scanning through. I saw all these cool things, right? As I'm doing it. That's how you set up your experiment if you really want to know. Sometimes you can put shortcuts in here. hey, today on the way home from school, I saw this and I really want to check it out. So I wonder if I do this, right? Here's my hypothesis. I think this is what I'm going to see. Do your experiment, set it up, collect your data, and then rearrange it. So will it be exactly the same as this if I take the letter E off of a magazine or out of a picture? When we look at like dollar bills, the way that they're printed and the material they're printed on is very different. So will the letter E on a dollar bill look the same as the letter E in the newspaper? Or will the letter E in a dollar bill look the same as the letter E in a magazine? What about those colors? Were the colors the same in a magazine and a shiny piece of paper as they were on a newspaper clipping? Test things out, play. Okay. We kind of already did the next page that says text analysis. So you look for the letter E, right? You draw your images, look at the edges of the paper, compare them. So draw a couple, this is a time for you to do and play as you want, but draw a couple circles and look at the edges of the paper. Look at the edge of the newspaper that was torn. What's it look like? Draw a little picture. Can you see the fibers? Is there a weave? Or are they just all mashed up together? Do the same thing. Rip the edge of your bookmark. Put it under there. See, is it the same kind? Is this like a plasticky material or is this actually paper? Right? Paper's just real thin, fine shreds of tree that are twisted or bunched or smashed together. You can actually look up making paper out of dryer lint on the internet. And they'll show you how you can take a piece of your dryer lint, smash it out, get it wet, and turn it into a piece of paper that's kind of fun to write on and make notes. You can do those different things. Ink analysis. This one's another one you can actually do on your own. We did it a little bit, but take the tip of your marker, put it on the different papers and compare them. See what happens. You can take a different color marker, right? Take a blue one, do the same thing. See if it makes a difference. Another fun thing is to take the marker and put it on a little edge of paper or a paper towel. So I put a little blue dot on, and then take some of your hand sanitizer. Everybody's got it nowadays, right? Take a little bit of hand sanitizer. Put it on a tip of a toothpick. Like this. Do it. Oh, got a lot there, right? And put that on it. See what happens. There's where my garbage containers indeed. See what happens. You can take that piece of paper or a different piece of paper, the same thing, put a little ink on it, down here. Another piece of ink here on the edge. And add some water and add a little cup. Oh, where'd it go? Just like that. You can actually put it down the edge of the water and watch it move up. So that's all wet. You can take it and put a slide under here and then investigate it later, right? So I can sit and watch if I wanted to, or I could take a peek, walk away from it and come back and take another peek. But it's really cool when you learn to use some of these tools, like for forensics or whatever, you can do analysis on fabrics. You can look, like in this little story about We the People, there was a guy that discovered, he bought a frame in a garage sale in Pennsylvania, Philadelphia, took it home because the frame was kind of pretty, but the picture was ugly. And as he was taking the back off it, he noticed there was a piece of paper in the back, and it was actually an original copy, which seems weird, but an original copy of the Declaration of Independence, right? So they didn't have Xerox machines or scanners back then. They had to actually write out every copy, and every person had to sign it. John Hancock had to sign every copy, et cetera. And so they could analyze the paper, they could date the paper, they could look at the signatures, the match, they could actually see the ink and look at the inks and dyes and everything, because of some of the skills that you learn with a microscope. And when we come back for the second one, we'll learn to make some slides, and we'll start playing with a lot of things with the slides. So thanks. I hope you're having fun with this so far. Take a break, take a breather, play with some things on your own, and then I'll come back and show you how to make slides and make your own experiments.